So today we're going to be talking about motility with a T, and we're also going to talk about micropipettors just so you have some experience at least seeing those and us talking about them. If you took Biology 150 before the spring semester, then you actually used them. I don't think we had a chance to use them in the spring before we left, but we'll get into that. First, we're going to do motility. So can everyone see the handout on motility? So we've talked about motility in lecture. Remember, it's motility with a T, not mobility with a B. So motility. And we're going to talk about two techniques for observing motility today. We'll talk about using motility agar deeps, as well as making wet mount slides. And then we'll see two videos actually looking at the live unstained bacteria in the microscope. And you will not get that wonderful experience of trying to find those unstained bacteria in the microscope and then trying to determine if they're motile or not, because it can be very challenging. But remember that not all bacteria are motile. If they are, they use flagella only, right? Cilia and pseudopods are for the protozoa, like paramecia and amoeba. But for bacteria, if they can swim, they use a flagellum. If they have just one, we call that monotrichous. If they have a cluster of flagella, we call that lophotrichous. One flagellum on each end is amphitrichous, and if they're covered with flagella, they're peritrichous. Remember, flagella are way too small to see. The only way to see them is by using a special stain, which makes them thicker so they can be seen. And we'll look at some examples of those slides in a few minutes. And we'll talk about some of the problems with those slides as well. So when it comes to the agar deeps, so the motility agar deep is a technique for determining motility. It works well for most bacteria, but not all of them, as we'll find out. But the idea behind motility agar is that it's softer than regular agar. It's less dense, and that can allow bacteria to move through the agar. Now, this technique does require steady hands. So we're going to use an inoculating needle, a straight wire, although you would normally have to straighten it out because they bend very easily. But you take that inoculating needle and you remove some bacteria from an agar slant, and then you very carefully stab the motility agar deep. And so the idea here is that you want to go straight down into the agar and then come straight back out. But the key thing is just to come out the same way you went in. If you go in at an angle, just come back out at that same angle. But you want to have a neat, clean stab. And the reason why, we're trying to see if the bacteria can move away from that stab. So that's why you need a neat, clean stab. So if you don't have a steady hand or you're not paying close attention, when you go down into the agar, you kind of move the needle all around through the agar before you pull it out. Well, the problem there is that you've now just spread the bacteria throughout the motility agar. So how do you know whether they actually moved away from your stab or you just spread them all over? So it was a technique where you had to be careful, where you needed steady hands. Now, one of the problems with motility agar is that it's already a little bit opaque. It's a little bit cloudy to start with because it's agar. Well, bacteria have no color by themselves. So as they grow, they just look a little bit cloudy, a little bit opaque. So how do you see opaque bacteria in opaque agar? It was very hard to do unless you have color. So there is a way to give the bacteria color. The agar contains tetrazolium chloride, or TTC. It is a chemical that turns red wherever the bacteria are growing, because the bacteria will donate electrons to the TTC, and when the TTC gets reduced, it turns red. So it just helps us to see where the bacteria are in the agar deep. So it's not pigment, it's not a dye, it's a chemical that will turn red when it gets reduced by the bacteria, so that's how we know where the bacteria are. So if the bacteria move away from the stab, that means they're motile. But if the bacteria stay in the stab, that means they are non-motile. So in tube A, the bacteria have moved all throughout the agar because it's cloudy red. In tube B, all the bacteria are still in the stab. So A is motile, B is non-motile. So any questions on that?
How did they discover that that chemical can, like, did they make it or was it a discovery?
I have no idea how they discovered that it gets reduced by bacteria, but luckily they did. Otherwise, it's very hard to see motility in agar without any color. So, but I don't know exactly how they discovered it. But in your spare time, which I'm sure you have a lot of, you could look that up, right?
Yeah, I'll do that.
Now, there are some problems with motility agar. Not all bacteria that are motile can actually make it through the agar. Particularly if they're a spirochete, if they're spiral shaped, it's really hard to corkscrew their way through the agar, so they just don't move. Also, if you had an organism that was an obligate aerobe, it's not going to like growing deep in the agar, because as we're going to learn again in lab next week, we talked a little bit about this in lecture, but oxygen cannot diffuse very well into agar. And so the agar deep tube is actually mostly anaerobic. So if you have an organism that must have oxygen, it's just not going to grow in the agar, so you're not going to get an accurate result. Almost any other kind of anaerobe is going to do just fine. Now, what if the organism is stuck to other cells? What if it's clustered together? Would you expect that organism to be motile if it's stuck to a bunch of other cells?
No.
Well, probably not, because it's hard to coordinate movement when you're stuck to a bunch of other cells. So if you have an organism whose name starts with staphylo, that might suggest to you it's most likely not going to be motile, right? Because it's clustered together. Or if, as you're working with the organism from the agar slant, you notice that it's very sticky and clumpy, again, it's probably not going to be motile, right? So there are some, clues that you can use to make predictions about whether or not your organism is going to be motile. So let's look at some of or we'll go over the results in the results table here. So Aquaspirillum petersonii, this is an organism which is actually very highly motile, but not in motility agar. It won't even grow at all. So even though it's very highly motile, we don't test it in the agar because it just doesn't work. Now, Corynebacterium xerosis is a sticky, clumpy organism. So would you expect it to be motile?
Nope.
Nope, and it is not. Right, it's kind of sticky the same way that Mycobacterium is sticky. Although they're different organisms, but they're still kind of sticky and clumpy. E. coli. Remember when we looked at some gram stain images of E. coli? And we know it's a rod, but what was its arrangement? Does anybody remember the arrangement of E. coli?
Was it in chains?
Nope, not E. coli. E. coli was actually single. So single bacteria are probably going to be motile just because they're not stuck to anything else. So they can swim around. And E. coli is very motile. Micrococcus luteus. Anybody remember what was special about Micrococcus luteus? Nobody remembers what's special about Micrococcus luteus? Nobody remembers? It had a special type of cluster.
Wasn't it in a tetrad or?
Right, correct. Tetrads, so groups of 4 cells. So again, it's clustered together. So would you expect Micrococcus luteus to be motile?
No.
No, and it's not, right? Now, Proteus mirabilis is a small gram-negative rod, and it's single. So it happens to be very, very motile. And then Staph aureus, Staphylococcus, would you expect that to be motile?
No, because it's in a cluster.
Right, so again, usually bacteria that are clustered are not going to be motile because it's just too hard to coordinate the movement. I'm not saying there are not any cluster bacteria that are motile, it's just that it's not common for bacteria stuck to each other to be motile.
I have a question. If the cluster was motile, would all of the cells need a flagella, or would only or one or a few need? Could they
No, they would all have flagella.
Oh, okay.
Yeah, I don't think there's a such thing as a leader and everyone's stuck to the leader. So again, here 2A, this is what E. coli and Proteus would look like. And 2B is what Staph and Micrococcus would look like. Now, Corynebacterium xerosis, we said is very sticky, so it's not motile, but it also doesn't really even grow in the motility agar. You usually don't see much of anything. You might see some bubbles where you stabbed it, but usually there's not even any red color. It's not cloudy. There's nothing. So the motility agar does not work for everything. And after you inoculate, you do have to incubate 24 hours to get the results. So, but any questions on motility agar?
Professor,
Yes.
yeah, the A and the B, which one do you say is motile again, please?
Of these organisms here, E. coli and
No,
Proteus. Huh?
the ag, sorry, the agar.
A is motile. A is motile. B is not. Does that answer your question?
Yes.
Now, when it comes to flagella slides, the problem with staining those flagella is that they are very brittle and they do break off easily. So when you look at a prepared slide of a flagella stain, actually the majority of bacteria on that slide usually do not show the correct arrangement because the flagella have broken off. So here we see Proteus, and with Proteus we see that because we have some naked bacteria, right? Those scandalous naked bacteria that have no flagella because they all broke off. But this cell right here is a good example. And so what's the arrangement of flagella of Proteus?
Is it peritrichous?
It's peritrichous because they're all around that cell, so it's peritrichous. But you will notice that almost none of the other cells in this image show that correct arrangement. And so that's the major reason why in lab I actually would just show you images like this on the computer screen rather than have you guys waste your time and my effort trying to go around and help you find correct examples. So normally I would just do it like this. I would project an image and then we talk about it. Because on the lab exam, you do not need to know which bacteria have which arrangement. You just need to know the arrangement when you see it. So we have peritrichous here. With spirillum, and this is true of both the spirillum volutans and the aquaspirillum, we see one flagellum on each end. So what's that arrangement?
Is that amphitrichous?
Amphitrichous? Amphitrichous?
Yeah, one on each side.
Yep. Here we have Treponema pallidum. So this organism is a spirochete. What kind of flagella do spirochetes have?
They're the ones with the axial filaments, the
Right,
endoflagella.
the axial filaments, the endoflagella. So you cannot actually see them unless you have something like an electron microscope anyway. But remember, they do not stick out. They are attached to the cell on one end, and they run along the cell body. And they're actually in between the cell and an outer structure called a sheath. And that causes them to twist like a corkscrew. So Treponema pallidum is actually the cause of a sexually transmitted disease. Any idea which one? Hopefully one with whom you have no personal experience. Any idea what Treponema pallidum causes? Starts with an S. You guys don't know your STDs or STIs? S. Syphilis. Syphilis. So what you're actually looking at here is a tissue prep. So what they did is they took some poor little animal, they infected it with syphilis, and then they had to, sacrifice it to slice up its tissue. And then they used what's called a silver stain, which makes the spirochetes black. And the tissue is a golden yellow or golden brown color. And so this poor animal was very heavily infected with treponema. It's actually very hard to get a hold of these slides today. They just don't make them anymore. So but fortunately we have the internet with all these images, although I do have maybe 2 old slides, and I'm talking like 40 years old or more, which I've kept and saved, which are still good examples because it's so hard to get these slides. Now the Borrelia is not shown here, but Borrelia is a spirochete, and the slide that you would typically see is a blood smear because it's in the bloodstream. And Borrelia burgdorferi is an organism that causes Lyme disease. Have y'all heard of Lyme disease before?
Yes, from ticks, right?
Right, yeah. It's transmitted by ticks. So if you like to go play in the tall grass or in the woods or something, you have to be careful because you might pick up a tick and it could bite you and it could infect you with Lyme disease. Because Maryland is a high Lyme state, so we do have to take precautions. Usually, Lyme disease is treatable with antibiotics like doxycycline, but you may not know you have Lyme disease until you have symptoms and then some damage has occurred because of the character. They always talk about that characteristic red bullseye that you get where you're bitten is actually not typical. Most people don't get that. And so you may not know that you actually had Lyme disease until a few years later when you start having all kinds of problems like arthritis and maybe some mental issues. Because I did have a student once, he was a former military because he'd been medically discharged because he actually had Lyme disease that went undiagnosed for a few years. And they finally diagnosed it because he had lesions on his brain. So, there are potential long-term consequences if you don't get, treated. And you can get tested for it if you're not sure. But normally that organism would just be in a blood smear. And it's a spirochete, so again, it has the endoflagella. Now, for the wet mount, and we're actually going to see some videos of this technique, the idea is to observe live unstained bacteria and see if they're actually moving on their own. Now, all particles suspended in water move. We call that kind of movement Brownian movement or Brownian motion, because the water molecules are hitting those particles and causing those particles to randomly vibrate. So there's always some Brownian movement present. In addition to that, are the bacteria doing something that appears purposeful, like bending, twisting, twirling, spinning, moving, in a lateral line in one direction, like they're actually swimming? And that can be very difficult to determine. So what we do is we take a slide and you actually draw a ring of oil using the oil immersion. In the center, you put a drop of water, and then you take a loopful of bacteria and you touch it to that drop. But there's no mixing here. You're just going to touch it. If you see something, stop. If you don't, touch it again. But when you see something, stop. And then you put a cover slip on top. Now, we only go up to the high dry lens, so only 400 times magnification. And the reason why we don't use the oil is because you have a cover slip sitting on a ring of oil, and if you put oil on top and try to put the lens in the oil, the cover slip is floating in between two layers of oil, and it just tends to move things around too much. So it's just too hard to use oil, so we just avoid using the 100x lens. Now, when you make your slide this way, you do need to make it quickly. And one of the reasons why is oil spreads out, and you want the oil ring to kind of be high when you put the cover slip on, because sometimes the water will simply travel over the oil ring and it will leak out, right? So you have a leak, and then everything is just moving towards the direction of the leak. And the longer that it takes you to look at your slide, remember the oil and water don't like each other, so they will start to fight. And that can make your slide very messy. So it's a technique where you need to make your slide quickly and then examine it and try to find your bacteria and then watch them to see, are they doing something purposeful? So you do have to play with the iris diaphragm a lot, because basically you'll keep it closed almost all the way, because too much light, and that's all you see is white light, because the bacteria are colorless, so they have no contrast. And you're focusing through water, so you just won't see anything except white light, so you got to close the iris. And since you're looking for unstained bacteria, it can be hard to find anything to start with. And so one of the things that often happens when you put your cover slip on is that you do tend to trap some air bubbles. But air bubbles are actually okay in this case because an air bubble is something you can see, you can find it. And then you focus on the edge of the air bubble, and hopefully on the inside will be air and on the outside will be your bacteria. And that will just make it easier for you to find something, right, by looking for an air bubble. And then you got to sit and you got to watch and say, okay, are these bacteria doing anything purposeful? Or are they just randomly vibrating? And it's not that easy to tell. When you look at something like Staph aureus, you know, those cocci are just bobbing around and vibrating like crazy. It looks like they're motile, right? It looks like they're moving, but it's all random vibration. But they really do look motile. So when you make your wet mounts, we ask the students to make 2 different wet mounts. Everyone makes one of the aquaspirillum because that's nice and motile, and you can see the corkscrews wiggling around. And then we ask the students to make a wet mount of the same organism they stabbed into the Motility agar deep. And the reason for that is so you can make observations of your wet mount. Do you think it's motile, or do you just think it's Brownian? And then you compare that to your agar deep results the next time and see if they match. Because sometimes students think, oh yeah, my bacteria, they're motile, they're moving around like crazy. And then their agar deep, it's just the stab is red. All right, so the bacteria actually were not motile. So we do like the students to do a little comparison of what they're actually seeing in the microscope and then compare it to their motility agar deep results next time. So any questions on the technique? So I just told you the aquaspirillum is very motile. You'd see it wiggling around. Acrinobacterium xerosis, remember it's sticky, it's clumpy, so you won't see anything except for the bacteria just sitting there. E. coli is motile, so you see them moving around on their own. Micrococcus, no, right, because they're clustered, so they just kind of vibrate around. Proteus mirabilis, remember Proteus, Peritrichus flagellus, so yeah, it'll be moving around like crazy. And then Staph aureus, staph, they're clustered, so again, it's just Brownian. Now, you would be working with agar slant cultures. So where do you think would be the best place on the agar slant to get your sample to observe motility? Where do you think would be the best place to observe motility? Using bacteria from the top half or from the bottom half?
I would say the bottom half.
So the bottom half is correct, because remember, that's where the youngest, healthiest bacteria can be found. It's in the bottom half. So that's where you want to get your sample, right, to observe motility. If you take the bacteria from the top half, well, those bacteria are old, right? They're sick, they're dying, so they're not going to be motile. So just something to think about, right? Just all these little things to always think about in lab. And because these are wet mount slides with live bacteria, you would discard them into a dish of disinfectant so we could then dispose of them later. So, in a microscope, how can you distinguish true bacterial motility from random Brownian movement? So you guys answer. Looking through a microscope, how do you know the bacteria are actually motile? What kinds of things might you see the bacteria doing?
It might be like bending or twisting.
Right, so bending, twisting, twirling, spinning, actually swimming laterally, something like that, right? Maybe moving into and out of focus. So there are things that they can do besides just randomly vibrate. In a motility agar deep, how can you distinguish motile bacteria from non-motile bacteria?
You have to see if like if the red color shows around the point where you stuck the needle in, if it's moving outward.
Right, so if the bacteria are motile, they will move away from the stab. If they're not motile, they will stay in the stab. And so number 3, what's the purpose of the tetrazolium chloride?
To show if the bacteria move by
Well, to show us where the bacteria are, right? It just helps us to see where the bacteria are. And why does the TTC turn red? Remember, why does the TTC turn red?
When it becomes reduced.
Right, so the TTC picks up electrons from the bacteria becoming reduced, and then it turns red. And what are the 4 arrangements of flagella in bacteria? What do you call it when they have one on each end?
Amphitrachus.
Amphitrachus. What if they're all over?
Peritrachus.
Peritrachus. If they have a cluster of flagella?
Lophotrachus.
Yep, and just one flagellum?
Monotrachus.
Monotrichus, right? Monotrichus. So now let's watch a couple videos. And neither of these have sound, so let's see if they'll work in Blackboard. Yes, surprisingly. Okay. So here we see staph epidermidis on the high dry. Now there is some drifting of the bacteria because there's a leak, but do you see anything that seems purposeful? And here we're seeing them under the oil. And you can see little clusters. So that's a hint, right? Clusters. Are these motile or non-motile?
probably not motile.
Right, so non-motile, what you're seeing is just Brownian movement, right? Random vibration with a slight current, moving to the right. Enterobacter aerogenes on the high dry. So what do you think about Enterobacter?
So in the other one, they were all moving in the same direction, aside from
Well,
being
that was just a current.
Oh, but here they're not moving in the same direction. So they're probably motile.
Oh yeah, look at these guys spinning and swimming laterally in one direction. I mean, they look like little insects, right? So it's clearly motile. And Enterobacter is E. coli's friend. They both live in the gut. And whatever this one is,
Is it motile?
oh yeah, those are clearly motile, right? I mean, look at those guys go. Again, they look like a bunch of little insects running around. If only it were this easy to determine motility. And then in the next video, they show you some of the techniques for making slides. Again, there's no real sound associated with this, no talking. So we do a modification of the hanging drop technique. Or you can also just use Vaseline, petroleum jelly. So any questions about the wet mounts or what motile bacteria look like? No? To point out that in the first one, we were looking at a phase contrast image for some of these, which is why it looks different. This one especially is a phase contrast microscope. What you saw in the other one is more like what you would see, just, black and white shades of gray. So, any more questions on motility? No? Okay, so next we'll talk about micropipettors. So there is this document with links on it. The first one did not work. Nothing was there, so I don't know. I don't think. I'll just check it again and see. Oh. Oh. Now it's working. It did not work this morning. Okay, so then we'll watch this one, but it didn't work this morning. It was just, black boxes. Nothing would work. So we'll watch this one to start with.
Micropipetting practice. Materials list. Micropipette, 1.5 milliliter tubes, 1.5 milliliter tube rack. Liquid, you may want to use food coloring in order to make it easier to see. Micropipette tips loaded into tip box. Waste disposal container. The micropipette is the tool of choice when pipetting small amounts of liquid, such as 1 milliliters or 1,000 microliters or less of liquid. The first thing we need to do in order to learn to use the micropipette is to introduce the parts. Important parts of the micropipette include the tip ejector or sidebar, the plunger for drawing up or dispensing liquid, the handle, the volume meter, which indicates the amount of volume being pipetted, and the tip holder. The first step to using a micropipette is to determine what amount of liquid you need to work with, and then choose the correct micropipette. Micropipette volume capacity will generally be written on the side of the micropipette. For our demonstrations, we will micropipette 100 microliters of liquid using the 20 to 200 microliter pipette. Set the micropipette to 100 microliters. Be careful to ensure you have the correct micropipette and that it reads 100 on the side. Once you've set the micropipette volume, you should open the 1.5 milliliter tubes you wish to transfer liquid between. Pick up the micropipette and notice as you hold it, it hangs over your index finger with your thumb gently resting on the plunger. Now you can put a clean micropipette tip on the micropipette. Warn your students not to slam the micropipette into the tip box. This will cause the end of the tips to bend and not be able to draw up liquid. Keeping the micropipette perpendicular to the liquid, draw up 100 microliters from tube 1 and transfer into tube 2. In order to do this, you will depress the plunger to the first stop. Place the tip into the liquid and then gently release in order to draw up liquid. If you've done this correctly, there should not be any air at the end of the micropipette tip. Maintain the micropipette's perpendicular position while transferring between tubes to prevent liquid from sloshing onto the micropipette. Once your micropipette is over the second tube, depress the plunger all the way down to the second stop to dispense the liquid. Do this slowly and gently. Keep the plunger depressed until the micropipette has been removed from the 1.5 milliliter tube. You can now eject the tip into the waste receptacle by pressing on the tip ejector. You may want to limit the number of tips available to students to discourage wasteful tip ejection. It is important to change your tip when changing solutions. Place the micropipette back on the stand when finished working with it. Just remember, accurate measurement of liquid can only occur when you depress the plunger to the first stop. Place the tip in the liquid and then gently release in order to draw up liquid. To dispense liquid, you will depress the plunger to the second stop and keep it depressed until the tip has been removed from the tube.
Boy, that was exciting, right? Yeah, this one's better. So in this next, it has some videos, it has a quiz, and you can see this morning we got 100%. And the people in the videos are British, and so they do not say pipette, they say pipette. So if it sounds kind of funny, they're saying micropipette.
Micropipettes are an invaluable tool used in many areas of science. They are used to accurately dispense a set volume of liquid with a high degree of reproducibility. They are available in a range of sizes from 1 microliter to 10 milliliters. This short presentation will show you how to correctly use your micropipette. In your work, you will need to use a range of micropipettes. Each one is easily identifiable by the label on the top of the plunger. The P20 is used to measure volumes of 2 microliters to 20 microliters. The P200 is used to measure volumes of 20 microliters to 200 microliters. The P1000 is used to measure volumes of 100 microliters to 1000 microliters. And a P5000 is used to measure volumes of 1000 microliters to 5000 microliters.
So the P20 used to measure 2 to 20 microliters, the P200 20 to 200, a P1000 100 to 1000, a P5000 1000 to 5000. Now of course there are different, pipettors besides just these. In fact, the ones that we use, our first one is actually 0.5 to 10 microliters. Our next one is 10 to 100 microliters, so there are slightly different ones. But the point is you should always use the correct pipette for the volume you're trying to pipette.
Micropipettes must be used in conjunction with the correct pipette tip. P20 and P200 use a yellow tip. P1000 uses a blue tip. P5000 uses a white tip. This is easy to remember as the tip colour matches the button colour on the top of the plunger.
Micropipettes are essential tools used extensively in laboratories today for the accurate measurement and dispensing of liquids. They are available in a range of sizes and can dispense anything from 0.1 microliters to 10 milliliters. However, they will only pipette accurately when used correctly. This short video will show you how to do this. It is important that the pipette is always held vertically to prevent liquid entering the internal mechanism, as this will irreparably damage the instrument. On the top of the micropipette is a volumetric guide that defines the maximum volume that can be dispensed, in this case 1000 microliters or 1 ml. The exact volume required is set by turning the barrel at the top of the pipette until the dial on the front reads the required volume. It is important never to attempt to dispense more than the maximum volume, as this can strain the internal mechanism and break the pipette. Firstly, a tip must be attached to the end of the pipette. Insert the end of the pipette vertically and press down firmly to ensure the tip is attached. Do not press too hard or the end of the tip will deform. The pipette push button has two stop positions. The first position can be felt when the push button is depressed to about 3/4 of the way down the barrel. In order to draw liquid into the tip, depress the push button to the first position only, insert into the liquid to be dispensed, and slowly release the push button to the start position. If the release is too sudden, there is a risk that air bubbles may enter the tip along with the liquid, therefore resulting in an inaccurate measurement, or the liquid will shoot up into the barrel of the pipette. To dispense the liquid, fully depress the push button to the second stop position. This extra depression ensures that the liquid is fully dispensed. The appearance of air bubbles in the sample indicates a correct procedure. Remove the tip fully from the liquid before allowing the push button to return to the start position. All tips must be disposed of appropriately, in this case into a discard jar. A firm press on the tip ejector button releases the tip.
So has anybody actually used micropipettors before?
I've used a pipette, but
Well, but the micropipettors.
150 class.
You did. So one of the things that you experienced, you know that when you push down on the plunger, there are two stops, 2 points of resistance. So as you're pushing down, you encounter the first stop. If you keep going, then you hit the second stop. And the first stop is used to fill the pipette. So you actually will push down to the first stop before you go into a tube, and then slowly release the plunger, right, to suck up your sample. And then when you put your sample into another tube, you push all the way down to the second stop, hold it, pull out of the tube, and then release the plunger. Otherwise, you will suck stuff back up into the pipette. So learning the first stop, second stop, and getting a feel for that was very important, because students had a tendency to always push all the way down to the second stop and then suck up their sample. When you go to the second stop and then suck it up, you're actually sucking up twice as much. So, you know, getting a feel for the different stops was important and took some practice. Of course, the ejector, you saw that, you push that down and it pushes the tip off. So you rotate this to set the volume, and setting the volume is important because you only have 3 numbers to work with. Even if your volume is, let's say, over 1,000, you still have just 3 numbers to work with. And so we're going to go through some examples of that. But if this is the P200 and it's set for 200, it's actually 200 microliters. If this is the P20 and it's set for 2 zeros, that's 20 microliters. So 200 is either 200 or 20, depending upon which pipette you're using. So that's why you have to know what those numbers mean. And it's also very important never to try and set the pipetter above or below the calibrated range. Otherwise, you'll damage the pipetter. Of course, the tip is what you push on, and then you push the ejector button to push it off. And a different color tip helps to tell you which pipetter it's for. Oh. is this? Write the quiz. I'm gonna have to retry it. Okay, so which of the following pipettes would you use to measure these amounts? So we're given some milliliter values, so we must convert them into microliters. So 0.15 milliliters, how many microliters is that? .15 milliliters is how many microliters? Remember, milliliter is bigger than microliter, right?
Maybe 20.
Remember, milliliter is 10 to the minus 3 liters. Microliter is 10 to the minus 6 liters. The difference between 3 and 6 is 3 decimal points, right? So we're going to move the decimal to the right 3 places. 1, 2, 3. So that would be 150 microliters. So which of these pipettors would you use to pipette 150 microliters?
The P200.
200.
Okay. So if you want to pipette 0.01 milliliters, so again, convert to microliters. How many microliters is 0.01 milliliters?
10.
Right, so remember, move the decimal over 3 places. 1, 2, and then 3 would be 10. So then, which of these pipettors would you use?
P20.
Okay. What if you have 3 milliliters? How many microliters is 3 milliliters? How many microliters is 3 milliliters?
3,000.
3,000. So which pipetter would you use?
The 5,000.
All right, the P5000. And if you want to pipette 0.8 milliliters, how many microliters is that?
800.
So which pipetter would you use?
The 1,000.
Right, the 1,000. So let's see if we're right. Yep, we're right. So same idea here. You're pipetting 45 microliters. Which pipetter do you use for 45?
Is it the 5,000?
Which one?
I said 5,000.
For 45 microliters? Remember, we're already microliter.
Okay, sorry. That's 200.
200. For 2,500 microliters.
1,000. 500.
Which one?
500. That's 5,000.
Right, the 5,000. For 8 microliters.
20.
And for 575 microliters.
1,000.
P1000. So is everybody following along with this? We're correct. So here we're looking at the different numbers, right, to figure out what they mean. So consider the 3 examples of pipette settings shown in the table below. Which of the following statements are true? So a P200 set as an example 1 would measure 35 microliters. So if you're using these numbers here, and it's a P200, and you set it for 0.35, is that 35 microliters? If this is a P200 and
I would
it's set
say
for
yes.
0.35, is that
I would say
35
yes.
microliters?
I would say yes.
You say yes?
Yes.
Anybody else say yes?
Yes.
A P20 set as an example 2 would measure 175 microliters. So these numbers here, if it's a P20, is that 175 microliters?
No.
No, because a P20, what's the maximum?
20.
20. And if it were set for 20, what would these numbers be?
002.
Now, if it's set for the maximum,
For the maximum 020.
no, 200 would be the maximum on a P20, 200.
Oh, okay.
So if it's 175, what is that actual amount in a P20?
0.5.
17.5 microliters. If you have a P5000 set, as in this example, is 175 1.75 milliliters? If this is a P5000,
Yeah, it would work.
how many microliters is 1.75 milliliters?
1,750.
Right, so 1,750. If this is a P5000, again, what would the maximum setting be? What would the numbers be for the maximum setting? If you're setting it for 5,000, what would the numbers be?
500?
00.
500, right? 500 is 5,000 on this. So if you set it for 4, it's 4,000, 3, 3,002, 2,001 is 1,000, and then 750. Does that make sense?
Yes.
A P1000 set as an example 3 would measure 10 microliters. So a P1000, if this was the setting on a P1000, is that 10 microliters?
No.
How much would this be on a P1000?
100.
100, right? Because if this were a P1000 and you set it for 1,000, it would be 100, and that would be 1,000. So 010 is actually 100. So that's wrong, right? This one here is wrong. And then a P20 set as an example 2 would measure 0.175 milliliters, a P20. So again, convert this to microliters. How many microliters is this?
That would be 17 and a half.
All right, so 17.5 microliters. On a P20, is this what you would get?
Yes.
Yes, right? So that should be true as well. So let's see if we're right. We are. So which of the following pipettes would you use to measure 0.15 milliliters? Again, convert to microliters. How many microliters is 0.15 milliliters?
150.
150. So which pipetter would you use?
The P200.
And if you had 1.75 milliliters, and you should have used a different number because we already saw that. How many microliters is 1.75 milliliters?
1,750.
Right, 1,750. So which pipetter would you use for that?
The P5000.
5,000.
5,000. Yep. So again, which pipetter would you use to measure 0.1 milliliters? Right, convert this to microliters. How many microliters is this?
Is it
How many?
100?
With the decimal 3, 1, 2, 3, so 100. So which pipetter would you use?
200?
And for 700 microliters,
1,000?
which one? What did I? Oh, I can't go back and see, can I? Nope. Oh, no, I don't remember what we clicked. So, which of these tips would you use with a 1,000 microliter pipette?
The blue one?
The middle one, the blue one.
Correct. Which of these tips would you use with the 200 microliter pipette?
The yellow.
Yep. Yeah. Well, our score was better than that, but I don't remember what we clicked on that one. So, but anyway, you can play with this on your own if you want. The document with the links is there. And then oh, and this is just an explanation. So, we don't actually do this. If we were in class, we could do this, but we can't do this here, so we're not going to. So, any questions on micropipettors? If you're going to go back and watch this again, this is a flash animation, so you have to activate Flash or enable Flash in your browser. So, any questions on micropipettors?
Professor, I have question on the exam when you're done.
Yep.
Yeah.
So what's your question?
Professor, the exam for 1 hour, I think it's not enough.
I know the morning said the same thing, but
Because
remember,
you have a lot of writing things to write, and eventually, like me, I skip most of the writing. Before I got there, the time was over. I was not,
well, your writing does not have to be perfect, but I understand. But remember, for most multiple choice questions, you should only spend 30 seconds to a minute on them. If you spend too long on multiple choice, there's not enough time for the written, right? But the first exam is always an unknown.
Okay, hopefully maybe the other ones will be better.
Right. It
Hopefully.
always gets better after the first one.
All right. Thank you.
But it's
Also,
the same
Professor,
exam.
you said
It's the same
the-
exam I give in class. All right, so I don't have anything else if you don't.